Have scientists actually created life? This video explores the cutting-edge fields of cellular synthesis, genetic engineering, and advanced lab techniques to examine the current state of synthetic biology and the milestones in creating minimal cellular systems. What does it mean to truly create life? We must distinguish between synthesizing an organism from scratch, cloning an existing one, modifying biology in a lab, or attempting to reanimate dead cells. Each of these represents a different tier of scientific ambition and technical difficulty. To understand the challenge, we define life through its fundamental characteristics: metabolism for energy conversion, reproduction to pass on genetic information, evolution through natural selection, and the ability to react and adapt to the environment. The theory of abiogenesis proposes how life might have emerged from non-living matter on the early Earth, through chemical evolution leading to the first replicators, eventually sparking the long process of biological evolution. A cornerstone of this research is the Miller-Urey experiment. By simulating the conditions of the early Earth, such as its atmosphere and electrical discharges, researchers demonstrated that the building blocks of life, like amino acids, can form spontaneously. The RNA world hypothesis suggests a vital intermediate phase. It proposes that RNA, acting as both a repository for genetic information and a catalyst for chemical reactions, served as the bridge between simple chemistry and the complex life we see today. Currently, the simplest artificial protocells we can construct involve lipid membranes and basic vesicles. While we have made strides in creating structures capable of simple mimicry, true autonomous self-replication remains a primary objective. A landmark moment occurred in 2010 when Craig Venter and his team synthesized the genome of the bacterium Mycoplasma mycoides. This achievement was a major step toward proving that genetic instructions could be synthesized and used to control a living cell. Was this truly an artificial organism? While the genome was designed and built from scratch, the experiment relied on an existing natural cell as a host. This sparks an ongoing debate about the definition of synthetic in biological creation. Today, synthetic DNA research continues to expand. We are moving from the simple synthesis of viral and bacterial genomes toward the capability to design more complex systems, with the ultimate goal of full control over biological architecture. The applications of synthetic biology are vast. From the medical production of insulin and vaccines to creating new metabolic pathways in industry for biofuels, this field is revolutionizing how we approach science and manufacturing. So can we create a cell from scratch? The answer is, not yet. We can integrate synthetic elements into living hosts, but the complete de novo synthesis of a fully independent cell remains a significant challenge for future research. The biggest questions in the field remain the origins of the first genetic information, the emergence of the first metabolic systems, and the leap to the first self-replicating organisms. Despite the challenges, we have achieved significant results. We have demonstrated the ability to design and synthesize bacterial genomes and successfully transplant them into living host cells, resulting in functional organisms. In conclusion, while scientists have learned to design and modify life, we have yet to unequivocally demonstrate how the very first life appeared on Earth. The search for these answers continues. This video is supported by a robust body of scientific literature. We have consulted various international sources to ensure the accuracy and depth of the information presented on abiogenesis and synthetic biology. For those interested in exploring these topics further, this bibliography lists key books that have shaped our understanding, including foundational texts like Nick Lane's The Vital Question and Paul Davies' The Fifth Miracle. We have also referenced essential research articles, such as the classic Miller-Urey experiment from 1953 and modern advancements by Gibson and Shostak, which continue to push the boundaries of current scientific knowledge. Additionally, our research utilized insights from reputable online sources, including the NASA Astrobiology program and publications in journals like Nature and Science, ensuring the information remains current with the latest discoveries. This diverse range of materials, spanning academic papers to popular science literature, provides a comprehensive overview of how researchers are attempting to unravel the complex origins of life. The field of synthetic biology is evolving rapidly. Staying informed through peer-reviewed journals and institutional reports is essential for anyone tracking the progress toward artificial cellular life. We encourage viewers to consult the works of scientists like Stephen Benner on the RNA world, as these resources offer deeper technical insights into the chemical foundations of biological evolution. The references provided here serve as a starting point for your own investigation into the fascinating and highly complex quest to define and potentially create life in the laboratory. Thank you for joining us on this exploration. We hope this video has provided a clear and well-documented look at the science behind the origins of life and the future of synthetic organisms. For further learning, please refer to the links and citations listed in the description. Continued research and public discourse remain vital as we unlock the mysteries of biology,